Hi, and welcome to today's program, How Do Paleoanthropologists Identify New Species?, which is part of our ongoing Hot Topic series. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown-haired woman in a purple shirt in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, a few housekeeping notes. This discussion offers closed captioning. You can turn them on or off via the CC button, which should be located at the bottom of the Zoom interface. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen, so we can sort through as many as possible. The Q&A time really flies by. The Q&A box is also where we'll share any relevant links during the program, so keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Sheela Athreya, and then I'll join her here to take your questions. So now I'd like to go ahead and introduce our speaker. Sheela Athreya is a professor in the Department of Anthropology at Texas A&M University, where she is a Chancellor's Edges Fellow and a Presidential Impact Fellow. She's a paleoanthropologist specializing in quantitative analyses and comparative anatomy of the craniofacial skeleton among Middle and Late Pleistocene hominins, with a view to understanding the evolutionary history of our own species, Homo sapiens. She has conducted research in Gujarat, Western India, surveying sites associated with the Toba volcanic supereruption, focusing on paleoenvironmental reconstructions of Western India before and after the eruption. She has also participated in the Narmada Basin Paleoanthropology Project in the central Narmada Valley in India, excavations at Lower and Upper Paleolithic sites in France, and at a Harappan early civilization site in Western India. Her current research focus is a project examining burials from rock shelters in central India, part of a UNESCO World Heritage Site, working with a multidisciplinary team to reconstruct early Homo sapiens evolution in India using a combination of morphological, genomic, and archaeological data. She also conducts research in museums throughout Europe, Asia, and Africa, looking at original fossils of mostly Middle Pleistocene specimens. Finally, her research aims to decolonize the study of our evolutionary history by moving beyond the out of Africa model and building regional models using local voices and data that are more appropriate for South Asia. Related to this, she teaches and publishes on critical issues of voice, inclusivity, diversity, representation, and post-colonialism in science more broadly, and biological anthropology specifically. And finally, Sheela was very recently named the 2023 recipient of the Robert W. Sussman Award for Scientific Contributions to Anthropology by the American Association for the Advancement of Science. So thank you for being here, Sheela.
Thank you so much for having me. Hello, everyone. It's a pleasure to be here. Thank you very much for coming, and to Briana and the National Museum of Natural History for inviting me. I am going to start my slides now and spend about 30 minutes talking about my research in human evolution, and in particular in the issue that we grapple with and we communicate to you is that in the public of how do we identify new species? How do these trees get formed? So as Briana said, I'm a paleoanthropologist working on the Middle Pleistocene, which began about 780,000 years ago. And I also work in the Late Pleistocene, which began about 128,000 years ago. And this is a critical time period for two reasons. One is because it's when our own species, Homo sapiens, evolved. And that's actually what I really focus on and what I'm really curious about. And during this time also, it is the first time really in our evolutionary history that we have populations throughout Europe, Asia, and Africa. And so we have these specimens in Southern Europe and Eastern Europe, in Northeastern Africa, South Africa, China, India, and in Indonesia. And these all date to the time period just before the evolution of our species. So what we name them is important, because if they're all separate species, then only one led to us. So in order to understand the evolution of our species, we really have to know how these regional groups are related to each other. And if possible, and did only one lead to us? And if not, then maybe the differences that we're seeing really just reflect something that we've always known about our species, which is that it's always been a really geographically dispersed and very physically varied species, that we've always been that way. And so this is how what we do really ties into what all of us think about our humanity, because how we identify a new species shapes our ancestral tree, but it really also shapes how we understand ourselves today. And even more so, it matters where we're drawing the boundaries of our species, because we're saying who belongs and who doesn't belong. And whether we realize it or not, we're policing the boundaries of who among us is more or less human. And how we deal with variation in the past does inform how we deal with variation today. So I'm going to be talking about how we name new species and sharing some of the fun stuff with you, but also sharing with you some of the things that we grapple with when we're trying to be mindful of these issues. So if you're here, you're interested on some level in human evolution, I imagine, and you have some understanding of what we do, which, of course, the funnest stuff is fieldwork. So this is one of my favorite human beings in the world, Dr. Rick Potts. He's the head of the Human Origins program at the Smithsonian and one of the best paleoanthropologists out there. So this is him doing fieldwork because it's just a better picture. But, it comes as no surprise to you that we do fieldwork, we find fossils. And between that and naming a new species, to some extent, it's fairly straightforward. We excavate, if we're lucky, we find something, and then we take them back to the lab. This is my team in India. So my collaborator, Dr. Ravi Koisadar, is just behind me, and then across the table from me are two of my students, Harshita Jain, who's a graduate student, and Urvi Kel, who was an undergrad with me. She's now doing her PhD in Connecticut. But so we take them back to the lab and we compare them. We get brilliant osteologists like a former Texas A&M grad student, Kristen Hofmeister. We do comparative anatomy. We look at everything and we fit the fragments together and we try to understand the morphology or the shape of what we're seeing. And we'll collect measurements on these specimens. All right, so you collect the data. You run some analyses, you shake it all up, and out comes, wait for it, wait for it, wait for it, new species. Okay, actually, that's not a new species. That's just me getting really excited about finding a frontal bone because that's my favorite bone. But, in the best of all worlds, Stanley Tucci makes you a negrito, and then you publish that you have a new species, right? But of course, there's a lot of stuff that goes in the middle, and some of it is stuff that you would expect. And some of it is maybe issues that you wouldn't have thought that we grapple with. And so I'll talk about both of those today. So the outline for what I'm going to talk about for the next 20 minutes or so is going from more specific to more general. So the specific methods that we use, I'll address some of the common questions we get. How do you deal with the small sample sizes? How do you deal with the fact that what you're trying to understand is extinct? And then we'll take a step back and say, okay, how can you name something as a new species? You first have to define the species. And so I'll pan out to how do you define species? And then I'll bring you back to some of your high school biology and Linnaeus and taxonomy. And then I'm going to get really meta. and we're going to ask, are species actually real entities in nature, or are they just mental constructs? And this brings us to understanding how our Western science fits in the global system of knowledge building. Is what we would identify as a species the same as what everyone around the world would identify? Would they name the same California redwood, or would they draw the same line at the boundary of Homo sapiens? And if so, then yes, we have figured out the real true structure of nature. But if not, then if they're mental constructs, then what does it mean when you have cross-cultural studies of who's human? Okay, so I'm going to start with the methods, or how did you get a species from that? And to be fair, usually new species are based and named on comparative anatomy. And there is a wonderful link on the Smithsonian Hall of Human Origins website where you can take an unknown fossil and you compare it to everything that's out there and you try to classify it. And you can get a sense of the most straightforward way in which we do our work. And the bigger the comparative sample, the better. We analyze it relative to everything out there that we know that can possibly be included. And if it's different enough from everything else out there, then it can't be included in an existing species and it's given a new name. So that's usually what happens. Sometimes a new name is announced because it's a reshuffling of other things. That may be because we have so much more evidence than we had 100 years ago when we first named a species that we have to revisit. And that's the self-correcting nature of science. And sometimes, like you learned if you attended Tom Hyam's talk last week, you can use unique genomic sequences to name a new population or to say that something's unique. So these are the straightforward ways in which we name a new species. And we communicate to you what we've got. The sample size question is a valid one, right? What's up with the small sample sizes? Like, are you kidding me? It's a tooth. How could you say that? How do we know? So that's a valid question, but to sort of put a finer point on it, how can you know that something is different enough to be a new species without knowing what the range of variation was in that species that existed in the past? You don't have a whole sample, a huge population size of fossils from that particular individual site. Usually you don't, to be able to say this one is typical of that and is so different from all the other samples that it should be given a new name. Well, to be fair, we usually do have more. These are some of the best case scenarios. So from the site of Cima de los Huesos, or the Pit of Bones in Spain, or the site of the Rising Star cave system in Homo naledi. So most of the time we are going on more, but regardless, it's a valid point, right? And we use the same approach that most scientists, and not just scientists, but statisticians use. The answer lies in statistics, right? We make the same assumptions that are made when people are polling about political candidates or political views, or when we're conducting medical trials to test the efficacy of something like the COVID vaccine. We draw a sample from the population, and you make an inference about the whole population from your sample. And in paleoanthropology, because we only have one, usually individual represented, we're making the assumption that that individual was drawn from the middle of this distribution, from the average member, and not one of the extreme outliers. And that's how, those are the assumptions that we rest our interpretations on. And, you know, the good news is that science is self-correcting, and that if we're wrong, as more data is found, we can refine these. So it's the assumptions that we make are resting on the statistical methods that are used throughout science. But also, how different does something have to be for you to name it a new species? Even if you're doing all of these appropriate methods, how different is different enough? And especially when we're looking at the Middle Pleistocene, where, as I showed you, there are fossil populations that are represented by specimens throughout Europe, Asia, and Africa. And so we know that on some level, it makes sense that they're going to be different because they live in different regions. And that's what we're like today. So how different is different enough to name something a new species? Because obviously today we look very different from each other, but we would never say that we're separate species. And so, we know that our physical differences are the result of adaptations to different climates and environments and different population histories and migrations and so forth. And that's where the understanding of how we define species and how we understand the evolutionary processes that shaped us are really critical to making an argument that we have a new species. We have to make a case, not just based on how it looks or how different it is, but on the evolutionary processes that we hypothesize are shaping that population. And so, like I said, when we're looking at this pattern of variation, we have to understand what shaped it before we compartmentalize and categorize everything separately. And sometimes separate species will look very different from each other and comparative anatomy is enough, but other times species will look a lot alike, and it'll be because of different habitats or different mating behaviors that they are a separate species. And so the strength of our names really does depend on understanding the whole ecological and environmental and evolutionary context in an evolutionary history in which our species evolved. And that is actually also very important for how we define a species in the first place. So up until now, I've been talking about a species, and the assumption is that you kind of know what I'm talking about, but wait, what is it actually? Let's dig a little deeper into that. Technically, as you know, if you remember high school biology, and so in Western society, Western science, but also most societies around the world, classify all the living organisms in their environment into hierarchically nested categories that go from most general to most specific. Our modern Western science has rules for determining how to assign organisms into these categories. And all systems are doing it based on shared characteristics, but in our case, it's everything that's a multicellular organism, and then everything that has a bony backbone encasing the spine, down to the genus and the species. And we belong in the genus and species Homo sapiens. And so here's just us. And the general belief, and what you're most likely taught, is that these higher levels of organization, it says here, are artificial, but it's not artificial in the sense that they're arbitrary and ill-informed. It's that they're artificial because they're based on our scientific judgment as to what should go together, based on the features that we think are relevant or salient or meaningful. But here it says species should also be real biological entities. And that means that they exist as a fundamental structure in nature, just the way that the trunk of a tree exists, or a molecule or a rock exists. Whether we've named it or not, it has it's got a real structure, and it exists in nature, and is one of the units by which nature is organized, right? So this is the idea that species are also foundational entities in the world. Whether we recognize them or not, they exist as real things. And so based on that, we just have to really uncover how they're defined. The most common definition that we're taught is that a species is made up of members of a population that actually or potentially interbreed in nature. And we call this the biological species concept. So if they can actually or potentially interbreed in nature and produce fertile offspring, then they're real species. And so if you can determine that you had gene flow and fertile offspring, then you can assert that you have found a new species or a member of a species. You can assign it to a species. But I'm sure as you're listening to this, you can think of so many exceptions right away. a couple of them that are relevant for human origins is like if something lived in the past, you can't observe if they interbred and produced fertile offspring, especially not as, before ancient DNA was able to be studied. Or what if you have evidence that they did interbreed, like Neanderthals and early Homo sapiens, but they looked really different and they lived in different places for most of their history. And gene flow was really just sort of at one time in their evolutionary history, and it was just in one place. So does that count as saying that they were actually always the same species? Or if it rarely happens or never happens in the wild, but it does happen like in captivity, say in zoos, is that a species? The point is, you can think of all sorts of ways in which this concept can't be applied by all scientists equally. And so to resolve that, there are other definitions of species that have developed. And they include the evolutionary species concept, ecologically, you can see them here, recognition, phylogenetic. The point is that each of these is based on a slightly different set of criteria. And what that means is that you end up getting very different evolutionary trees. So technically, each one of these trees of human evolution is correct if they're faithfully and consistently applying a particular definition of species to everything in the sample. And so to the outside, I know this looks confusing, but these are all scientifically supported. What's different is that each one of them emphasizes a different dimension of the very complex process of evolution. And so it's just emphasizing a different dimension of that to determine what's a species. Is it that they lived in the same ecological environment? Is it that they had gene flow? Is it that they have the same morphology or appearance? Do they stem from the exact same evolutionary lineage? So these aren't the product of just sort of personal disagreements or random internal disagreements or selective attention to facts. And I wanted to say that because it's really important to know that so many of the disagreements or differences and different interpretations that you will read about or hear about in paleoanthropology are not for arbitrary reasons. They stem from the very difficult and differential and complex ways in which we're trying to understand evolutionary history. And I just felt that was important to say because we're in a time when the idea of facts is really undermined in our public conversations. And so all of these are correct, and they are not just selectively based on, a selective attention to facts. But for a practitioner like me, yeah, it's really hard. It's really problematic, right? Because I like everyone, I'm trying to understand the evolutionary history and the origin of our species. But if some of my colleagues see Neanderthals as a separate species, and understandably so, because they see a lineage and they see different morphology and a different ecological niche, based on that evidence, I completely agree. But if for me, what really matters is that there's evidence of interbreeding and fertile offspring, then we're both going to be right. And it's not going to be more data or, better definitions of species that's going to get us to reconcile this. They're both valid ways to interpret our evolutionary past. But of course, it also does lead to very different histories of our species. And in particular, like I said, very different understandings of the origin of our variation, which is what I really care about. So it really does matter to get this right. And so if there are multiple species concepts and they're all scientifically grounded, how do we know what we're even talking about? Are they even real? And so this brings me to the final question that I am going to cover today, which is that to answer whether or not species are even a real construct, it is unfortunately not going to come from somebody winning the argument about what's the most important part of evolution. it's going to come from philosophy, actually. It's going to come from understanding whether they are, in fact, real natural entities in the first place or constructs of our mind. And this is a debate that has actually gone on since at least the time of Aristotle. So one of the bases of Western science is that the essential structure of nature can be known, and it is our job to just uncover these groups that make up the structure of nature. And this goes back to Aristotle. And for most paleoanthropologists, I would say almost all paleoanthropologists, species are real to them. I mean, they understand that there's variability in how we define it, but they would, I don't know, I shouldn't speak for anyone, but I know that we as a field do not communicate to the public that we are dealing with anything other than real, true, evolutionary, real historical entities, and that we are uncovering the truth when we announce a new species. One of the things that this does is it inherently elevates Western scientific methods above other forms of dividing up and naming the natural world. And so I think that one of the ways to untangle our confusion or our sort of dilemma is to look at how other societies do this. And this is the field of ethno-taxonomy. And what's interesting is that Linnaeus built his taxonomic system on pre-existing ones in Sweden. So when we say that, there's Linnaean taxonomy and folk taxonomy, Linnaeans is a folk taxonomy. They're all, forms of ethno-taxonomy. And ethno-taxonomy today is primarily studying indigenous and non-industrial societies to see how they name the natural world around them. What researchers have found, and I want to shout out to my colleague Allison Hopkins here, who taught me a lot of this, because this is what she does. But what researchers have found is that almost all societies have taxonomies, and they divide nature up in the same, most of them are hierarchical, from most general to specific. And the most common number of categories across societies is 5. And so that includes like all living things versus, rocks and mountains. All living things are classified. And then all things that are sort of grossly morphologically similar are shared. And so that would be, all trees or all fishes, and then so on. And the criteria get to more minute details. And so one point of sharing this with you is to say that, yes, on some level, all humans are perceiving the same gross patterns in nature, right? Not everything is just highly variable and open to interpretation. But also, it reminds us of one other thing, which is that the function of a taxonomy and these classifications and these labels and these names, it really actually isn't to win the contest over figuring out the real nature of nature. What it is supposed to do is to store and communicate information about the world around us to our fellow community members. So it's doing a lot less heavy lifting if you look at it that way. And I love this example of a study of the taxonomic categories among indigenous Hawaiians. One of the reasons that I got really into this is because I read a translation of a Czech biologist's book on his research in Papua New Guinea. And at the very end, he just mentions how they divide and name birds in the community he was working with. And more recently, a scholar has reclaimed native Hawaiian taxonomies for birds. And what you can see is that here, for example, this is from Malo's bird folk taxonomy, which was in the 1700s, developed in the 1700s by an indigenous Hawaiian, but sea diving birds belong in one category and fresh and saltwater and pond birds belong in another. And this has to do with the function of those birds within the taxonomic, within the subsistence and ecological and even economic system, and how they sort of perceive everything to fit into the ecological space around them. And it was very similar with the New Guinean community. And what's relevant to them is not whether, deep sea birds or pelagic birds evolved monophyletically from, like the smaller wild birds. They're using what's salient and important to them to create these divisions. And we are arguably doing the same. What's salient and important to us is to understand evolutionary change. And so if we put our taxonomic system and our practices into this cross-cultural framework, there are two takeaway lessons. You could get all these names that I have here on this slide. But really one thing I hope for you to take away is that when we do all the things I told you about, when we analyze the data and measure features on the skeletons and run the statistical analyses and decide that we have enough to name a new species, we're engaging in culturally specific practices. So we're using methods that mean something to us, that help us build knowledge, that answer questions that we care about, like I said, namely evolution. And therefore, we're identifying qualities in the fossils that we're looking at that are salient to us to help us answer this. And other societies creating taxonomies for other purposes would draw the lines very differently based on their needs. And both of these, and this is important, would be repeatable and testable and falsifiable. And so they're both forms of scientific knowledge building. And Linnaeus' taxonomy is really one ethno-taxonomy. The other thing that we've learned from these cultural studies is that we're trying to get at everything, trying to get at how many species lived in the past, right? And one thing we can all agree on is that everything from this time period that I showed you in the Middle and the Late Pleistocene belongs in the genus Homo. That's not under dispute. So everything in Europe, Asia, and Africa belongs in the same genus. Whether or not it's a separate species is debated, and that whole thing, that debate is really normal, because another thing that ethno-taxonomy has shown us is that the genus is considered the so-called building block of folk taxonomies. That's because the human capacity for pattern recognition at a consensus level happens at the generic level. And once you try to parse differences using more minute criteria, the more problematic they get. And even in other societies, there are these disagreements. And what tends to happen is that experts, so people who specifically work with one plant because of medicine or because of food or subsistence or whatever, tend to over-differentiate. They tend to see more species, or I should say, they tend to create and name more categories at the specific and varietal level than other members of their own community. And so non-experts in a community tend to just work at the generic level and name everything at the generic level. And so the lumper and splitter debates are really common across societies. They're not a product, again, of personal differences or bad science. Okay, so I was thinking about, I could wrap this up by repeating what I told you about the methods and the species concept and the ethno-taxonomy. But rather than wrapping this up by reiterating how paleoanthropologists identify species, I actually want to share a personal reflection. And that is that I became an anthropologist because I wanted to understand why we all look so different. And my dad's listening now, and so I want to shout out to my dad. And my parents would take me to JFK Airport, and child of immigrants, we'd go to JFK Airport a lot. I would see how different people looked. They had, National Geographic and Time Life books. We went to Smithsonian all the time when I was growing up. There was a face wall. And I really wanted to understand why we all look so different. But when I began my studies in graduate school, the models and the interpretations and the assumptions didn't always make sense to me. And they were things that I was being told that I could take for granted as starting points. But the fossils that would be being assigned to our species and called modern, to me, really all converged on one ideal type of human. And we were taught that these are the qualities of anatomically modern humans. But to me, it looked like a Northern European. And it made me wonder, what if Indigenous Australians were doing the naming and doing the phylogeny tree building and the classifying? Where would our species boundaries be drawn? And so, in other words, whose voices are not being heard in this conversation? And so now I'm lucky enough to have a platform where I get to have my voice heard. And so I wanted to take advantage of that by warning against what happens when we don't have a diversity of voices. What's happening now that I see in paleoanthropology is that we are inadvertently really reproducing the anti-Black, the anti-Semitic, the indigenous erasure that characterized early biological anthropology. And so there are inherent racial typologies and hierarchies in how we name and how we communicate hominin species. And I don't think we really realize that. But how we're doing the naming and how we're depicting the evolutionary history of our species really matters for how well we can make an argument against the thing that we all care about the most and agree upon, which is that biological race isn't real. So the models that we engage with and that we communicate to the public convey to you that there is that there's an origin of anatomically modern humans, and it was originated in sub-Saharan Africa and it's spread out, and that there's this one modern human form. And so that the program of paleoanthropology is related to find the first appearance of this of Homo sapiens around the world. And that's what I thought my job was when I first became a paleoanthropologist. But at the same time, this always sounded a little off to me, because it sounds a lot like the retelling of the story of European colonial expansionism, where a single form of human was adaptively superior to everyone else, and it spread throughout the globe and conquered everyone else. And using terms like modernity and searching for evidence of culture in things like art and music and technology are using very colonial criteria of what in order to police the boundaries of who gets to be called Homo sapiens in the past. So going forward, all of this is to just really reiterate why I think it matters so much that you understand all that goes into how we name species, because it really does affect how we understand variation and humanity today. And just the scientist in me argues that we have to get a better grasp of evolutionary processes that shaped us before we can speak with such certainty about how real these species were in our past. And so I want you to, going forward, if you take anything away from this talk, next time you read a news article, just take a hot minute to think about what the new name reflects. Consider that our names are a product of our cultural scientific processes, and that other societies would divide variation up differently. That there is variation there, but then there is also something underlying and real about the cognitive processes and patterns by which we all categorize and classify. And so it's definitely something that we can do and can reach a consensus on, but maybe not at our first pass, and maybe not with the smaller sample sizes that we usually have. So as you go forward, I hope that you think about whose voices are being reflected in these taxonomies and phylogenies, and also to understand that they're theories, that we test them, and that we'll continue to self-correct them as we get more data. So thank you so much for coming.
Thank you, Sheela. That was wonderful. And we have a bunch of questions already, so I'm just going to dive right in if that's all right.
Absolutely.
All right. So the first question, and you touched on this a little bit, but I think this is a great direct question from Stephen. If you can breed with it, is it a different species? Denisovans, Neanderthals, etc.
So that's the one, Stephen, that I always default to. And if you want me to defend why, I can. And that's where sort of our judgment as scientists comes into play, where we emphasize different aspects of evolution in order to understand variation. And I have just always emphasized, been focused on the role that gene flow has played in shaping or preventing speciation. And so that's where I would go. But I absolutely also see the scientific strength of a morphological species concept that would name Neanderthals, for example, as a separate species. I think that they're both valid, but I focus on gene flow. That's, to me, where I land.
Thanks. Here's an interesting question, talking about morphology from Steve, different questioner. Seems like species in paleoanthropology are based on the selection of holotypes and paratypes. How do you determine what constitutes a holotype and a paratype?
I can give you the technical answer, but I'd rather give you a little bit of historical background. And so you're right? So there are things called holotypes and paratypes, and a holotype is the type specimen of a fossil. And so it's the first specimen that was identified as being unique enough to name a new species. And then everything that comes after that is compared to the holotype to see if it also belongs in that species. So technically you're right, and that is part of there are rules, the International Code for Zoological Nomenclature. It tells us how we are allowed to name a new species, and there are very strict rules. Now, the thing is, Stephen, that those rules were made before we understood the complexity of evolutionary change. And so they really haven't kept up. So when Linnaeus was doing his building of taxonomies, he was, in his mind, classifying things in a fixed and unchanging world created by God. And so there was a type specimen, and that was your ideal. And it was just the perfect representative of this. So it's anti-evolutionary in the sense that there is no ideal type of any organism. And when you start talking like there is, you start getting into the whole question of purity, and variation is just, a problem, and it's noise in the system, when it's that range of variation that's actually the norm. So yes, that's what we use, but it's part of the problem because we're trying to understand evolutionary change and reject typology in living humans and reject race while still using a pre-evolutionary, very typological system.
Thank you. That's really helpful. Here's an interesting question from Dennis. He said, can you get back to the third or fourth slide? And we can do that. So you don't need to do that, but we can pull the slide up. He said, there's a ghost population. How and what do we know about this ghost population?
So this is an homage to one of the scholars who's been doing so much work on this, Dr. Chris Stringer. He's always been so wonderful and kind to me. So I wanted to include one of his diagrams in here, but this was really not meant to depict my view on anything, or again, it's not meant to depict what I think are objective realities. This is his interpretation of what the evolutionary history of humans might look like. And he's done a lot of work trying to understand the same time period that I've sort of, standing on the shoulders of giants. I built a lot of my work on what he's done. So, I don't see it the same way as him, but I couldn't do what I do without him. And so I really wanted to include one by him.
Thank you. You mentioned the idea of race a couple of times, and does it exist? Does it not exist? What does it mean? And so here's an interesting question from Sophie, who asks, is there any evidence that other species in the Homo genus had what might be termed races? And maybe we should just unpack what that might mean.
Yeah, I think because biological anthropologists, I think, invented the concept of race. It's a historically mediated phenomenon. It's kind of like inventing the flat earth, right? Like we invented, like geographers really can't go around anymore saying, oh my God, I've completely proven the earth is round. Okay, because no one ever, we know it was bad geographers 800 years ago who said it was flat, and now good geographers are reminding us that it's round. Similarly, bad biologists, 400 years ago created this concept of race and partitioned human variation out into these really, really for the purpose of racism. I mean, the history of race is the history of racism, right? For the purpose of being racist and ranking humans. And so it's not real. So no, I mean, it's not real in today, and it's not real in the past. Variation and the ways in which we varied are interesting and meaningful questions to ask, though. And so I think that you're right in that the way we varied in the past isn't necessarily going to be the way we vary today. And that's something that I would be, I want to learn and understand more about.
Cool, thank you. Here's an interesting question, maybe about the future. So yet a third, Stephen, asks, the concept of Homo species often implies that the process isn't static. We have Neanderthal genes and Denisovan genes, so we do, because there wasn't enough time for speciation to complete when they began to interbreed. If more time had elapsed, would there have been more than one human species?
Oh my gosh. I mean, there may have been more than one human species, right? Like, I understand those arguments based on certain types of data. I think that's possible. But like I said, because we have evidence of gene flow, I go with, incomplete speciation. We're all the same. They're all Homo. We've all just always gotten along, right? That's how I deal with that data point. But I was, but plenty of my colleagues who are just as rigorous about how they deal with this data legitimately say, look, you can have occasional gene flow, but they're still separate species. And there are examples in nature where that happens. For example, these baboons in Ethiopia. And so that's where it comes to, what are your criteria for naming a species? And is it separate evolutionary lineages that have 5 million years of separation? So even if you can interbreed occasionally, it doesn't mean you're the same species. Or if you can interbreed occasionally, all bets are off, same name. For example, if you're interested, there is what I'm talking about is a couple of species of baboons in Ethiopia, and we have a good fossil record for one of them, so good that we recognize that it actually goes in a different genus than other baboons. Briana, please correct me if I'm wrong, because this is your area of expertise, some Therapithecus, but today they do interbreed with neighboring baboon species in a hybrid zone occasionally. But the fossil record is very clear. 5 million years ago, at least, this species of baboon separated and has been on its own trajectory. So both are right. Again, evolution is so complex that you can't come up with a definition that accounts for all of the multiple dimensions of it.
Thank you. Here's a question from Urate who says, but folk taxonomies are not interested in species origins, but rather their social utility, which is not at all the same lens or objective. So their comparison can't be equated. Can you comment?
Well, we're maximizing what matters to us, and they're maximizing what matters to them. And that's my point. So the program of Western science doesn't trump other agendas, right? It's just the one that matters the most to us.
Mm-hmm. Here's an interesting question from Bram. Excuse me. Are the factors leading to diversification or speciation different in humans, in the human lineage, compared to other animals? For example, diverse cultural traditions might lead to diverging patterns of natural selection.
Yes, and I think that especially because we are such a geographically dispersed species, the one-size-fits-all model of what differentiates us is really unhelpful. So yes, in some regions of the world, at some points in our past, the cultural buffers may have provided a different role for selection to play than, say, genetic drift. And in other regions, selection may have played a stronger role, or isolation and drift may. And so we can't say that one event shaped all of living humanity. And that's why my work in South Asia and Southeast Asia is trying to elevate that data to be more considered, because in China, for example, in northern versus southern China, even, you have clearly different roles that geography, that climate, that migration played in shaping the populations there than it did in shaping absolutely contemporaneous populations living, say, in southern India at the time, or in Northeast Africa. So the one-size-fits-all model doesn't work. And yes, culture and other factors would have played a role. Regional differences are very interesting, and regional histories are very interesting.
Awesome, thank you. So Laura asks, would you like to say more about your research of the before and after Mount Toba eruption, especially the theory of a human bottleneck?
Oh, that's actually not my area of expertise. I'm an educated consumer of that research. But with one of my colleagues in India, Dr. Rachna Raj, who is, just as a plug, the best geologist in the world, I think. She found a place in India where there was a primary deposit of Toba ash. And I think that the role that the eruption played in shaping the landscape is unquestionable. But again, it wouldn't have had the same effect everywhere around the world. Also, I know that the pattern of population variation today does indicate that there was a bottleneck at some point in our history, but I don't, again, think that everyone everywhere can just came close to disappearing, right? And especially when you take into account physical geography and the parts of the world that I work in, the tip of India, India is a giant peninsula. And so the southern half of it is going to be very differentially affected, either by population collapses or by gene flow or demographic changes than the northern half of it, which is where you have more inflow and outflow. And then likewise, Indonesia, where I've done some work, is an island. Well, it's where the eruption happened too. But these southern hemisphere regions and their histories are not weighted equally yet in global models of human evolution. So I think with more information and more data points from the South, what was that bottleneck? How deep, how much did it affect everyone everywhere is going to come to light.
Thanks. This is a little bit following on that, and maybe just a sort of broad overview question, but excuse me. Roger asks, what are the methods for using DNA to determine species, genera, et cetera? And I know that your expertise is in the fossil record, but maybe just kind of a general overview.
Yes. And so, I'm guessing that Tom Hyam would have talked about this a little bit last week, but it is not unlike how we use comparative anatomy, right? Even with the Denisova specimen, it was one individual. And you may have a whole nuclear genome, but you have you've reconstructed decent coverage, but of one genome, one individual's genome. So it's all comparative, right? And we try to get comparative data from individuals that were from around the same time period, because it doesn't really do us as much good to compare someone living 30 to 50,000 years ago and see how similar or different they are from tons of people's genomic sequences today, because that ignores 50,000 years of that evolution has had in shaping genetic variation today. And so it's about comparing the unknown to the known and making very intentional and intelligent choices with your comparative sample, because that's really what determines whether you have a solid foundation for saying this was different enough. It was different enough from the things I should be comparing it to that lived at the same time,
Yep.
not from what I have in front of me. So
Great. Thank you. That's really helpful. All right. I think we have time for probably one more question. So I'm going to end with a question from Dirkira, who first says, thank you for sharing your experiences and scientific findings. I thoroughly enjoyed your presentation. Will you discuss your favorite expedition and your most memorable find? I heard you discuss being in awe about one of your favorite discoveries. Will you please tell us more?
Yes. So I hate to say it because I know you want a good answer, but I love every single time I go in the field. I love it equally. I feel so lucky. So I never get sick of it. And I'm one of those people, Briana, I wonder if you're the same, but like some people, you know, like I said, I really want to know why we all look so different, but I had a very circuitous path of studying film production and this and that. And what happened was that anthropology just never bored me. It was sometimes on the back burner, but it was the one thing that still doesn't bore me. I love it. I love studying fossils, and, you know, my mom was a radiologist. I love the human skeleton. I never get sick of it. So anytime I get to touch a fossil, I'm jazzed. But probably the most meaningful one for me was when I first got to study the fossil from the site of Narmada in India. So it's a hominin fossil. It's the only one from the Middle Pleistocene in South Asia. And it sounds cliched, and I'm almost like resisting saying, oh, I'm Indian and it was in India. But it really, I study our roots. It mattered to me that that was the one of the very first fossils I ever studied and held. And you know, one of the things that my advisor and I share is, and I think a lot of us shared, but he articulated, is like, these were people, these were humans that lived, and we are telling a little bit of their story. And I think that studying the Narmada fossil at the Geological Survey of India headquarters in Calcutta, gotten to go a few times, and every time I hold it, it really, my heart skips a beat.
I concur that every time I get to touch a fossil, I'm jazzed. So I get that. So thank you so much, Sheela. I'm going to wrap up today's virtual program. That was wonderful. Please join me in thanking Sheela for sharing her work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Ryan McRae from George Washington University, to our donors, volunteers, and viewers like you. And finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. Thank you. I hope you'll join us for our Hot Topic program next month on Thursday, November 17th at 11:30 a.m. Eastern Time with Dr. Keneloe Molapiane, a postdoctoral research fellow at the Center for Exploration of the Deep Human Journey at the University of the Witwatersrand in South Africa. She has excavated with the team in the Rising Star Cave System in South Africa, where Homo naledi was discovered. Check back on our website for more soon, including information about that program. We've put a link in the Q&A where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. It's the best way to stay informed on upcoming programs and learn more about the museum's research and exhibitions. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to our participants, to Dr. Athreya, and to you, the audience. See you next month.